Our lab investigates the molecular mechanism of synaptic plasticity under physiological and pathological conditions. And after I saw that I'm the next speaker after Professor Tsai, I decided to share with you some new finding that we have on the more pathological side, or more promising in a way, and describe work that we have using hyperbaric oxygen therapy to treat Alzheimer's disease. We heard about Alzheimer's disease. So what is hyperbaric oxygen therapy? So hyperbaric oxygen therapy, or HBOT, is a medical treatment while you administer 100% oxygen instead of the 21 that we usually breathe, but under a pressure that is basically above 1 atmosphere, usually 2 atmosphere. So patients are situated in a chamber, a submarine-like chamber with gas mask, and the pressure goes to 2 atmosphere usually. They stay there for 1 hour a day for a period of 1 month or 6 months or 2 months, depends on the necessary. What it does physically, it increases the solubility of oxygen in the plasma, and that causes something like a 5-fold, 6-fold increase in the solubility of oxygen in the tissue. A couple of years ago, Professor Shai Frati from Asaf Harofeh started to treat a patient that suffered from diabetic, and usually these patients have problem with wound healing due to low circulation of blood. So when they enter the chamber for a month of treatment, 1 hour per day, usually their wounds are healed. But what Shai noted is that some of them, before they entered, they had stroke half a year before it. And after this 1 month or 2 months of treatment, they came out almost like new. So some people that couldn't speak came out of the chamber while they can speak. People that couldn't move their hand could move their hand. And at that point, Shai decided, okay, this might be, by chance. I should stop it, or maybe investigate more. So he performed several more studies, and he showed that the stroke patient, even half a year after the stroke, after 2 months of treatment, came out with a significant neurological improvement. He also continued to examine people that had a traumatic brain injury with very hard cognitive impairment that suffered from that for years. And after these 2 months of treatment, they came out with very improved performance. At that time, we created contact with Shai, and as scientists, we were a bit skeptic of what he was telling us. And we asked about the process, about the mechanism. He couldn't answer something very scientifically based. And we decided to join forces and to try to understand the mechanism of HBOT, and also to try to see whether we can also help additional neurological disorders. And the idea was to try to see whether HBOT can serve as a valuable therapeutic practice to other neurological disorders that exhibit hypoxia-like pathology, like Alzheimer's disease. And basically, the aims was first to understand the mechanism of HBOT, and also to see whether what will be the effect on AD mouse model. So is there a link between hypoxia and AD? And yes, there is several indications that hypoxia may emerge in the brain of AD patient due to reduced cerebral perfusion. We know that hypoxia itself contributes to the accumulation of A-beta and phosphorylated tau. And also we know that there is a dysfunction of blood-brain barrier, and this also can lead to degeneration. And that also, following the cerebral ischemia and stroke, the incidence of AD and vascular dementia are increasingly increased. At that time, Ronit Shapira started a PhD in my lab, and we decided to move the clinical studies into the more mouse environment in which we can learn a bit more about the mechanism. We joined forces with the Israeli Navy in Haifa, and they built for us a small chamber for mice. And this is high quality, so they are coming every half a year to check that everything is working, like the chamber that are used for a diver. So it's we cannot go into it; it's too small, but it's very well preserved. So what we decided to do is to expose 2 mouse line, the 5XFAD that we just heard about, the triple tetanic mouse, for 60 minutes a day for 100% oxygen and 2 atmosphere. And after this treatment to perform either behavioral, physiological, or pathological condition, it's still in progress. I'll show you some of the results. The first model that we use is the same model that Louis showed before, the 5XFAD model, which have many of the Alzheimer's disease phenotype. Specifically for us, it was also important. This model also presents abnormalities in microvessels after near the A-beta plaques, and this suggests the impaired blood flow to this area. So the first thing that we did in these mice, we took 2 ages, either 4 months, which is shortly after the appearance of the symptoms, or 6 months, where you have the whole symptom already there. We had 2 groups in each one of them. Usually we treat them for 1 month, for 5 days a week, 60 minutes per day. And the control group just went into the chamber with normal baric pressure, normal baric oxygen, and then we performed several tasks. So first thing that we checked is whether there is really reduction in hypoxia state, and actually whether there is any hypoxia in these mice. And we used the hypoxia probe, and you can see the wild type, or the, these are wild type mice, and these are 5X FAD mice. These ones are without treatment, and what you can see here, that if the fluorescent level is high, meaning that these mice suffer from hypoxia, it is quantified here, and you can see that there is much more hypoxic state in the CA1 and CA3. And this is before treatment, and after the treatment, again, of 1 month treatment, there was a reduction in the hypoxic state in both areas of the hippocampus. It's also important to note that this was not done immediately following the treatment. This was done 2 weeks after the treatment. So it's not an acute effect of the oxygen that was still there, but this treatment started some mechanism that we'll elaborate in a minute, that kept this area under a normal hypoxic, non-hypoxic state. So this is for hypoxia. We then moved to look for a different behavioral aspect, and we checked the fear conditioning test, in which, in this test, the mice is put in a chamber. It's giving a shock. Usually a day later, when they enter the chamber, they freeze, since they recall the context. And 5X mice usually freeze less, and this is shown here. So they are less associating the context with the shock. Following the treatment of the HBOT, there was a significant improvement in freezing, suggesting that they could learn much better than before. What about the pathology of A-beta and additional markers? We checked first for the A-beta, and this clearly can be seen from the upper panel, which showed the 5X FAD mice, and this is after the treatment. So there is a dramatic, significant reduction in both the number of plaques, the area of the plaques, and the size of the plaques. At that point, we wanted to check whether there is also effect on neurogenesis. That is, we know that is also altered in AD. And in the Dante gyros, there is a continuous generation of newly born cells, and we can track them and mark them with specific markers. For example, the one that we use is double cortin, that is started to be expressed in a week or 2 weeks old neurons and kept for 2 or 3 weeks and then disappear. So we checked for the appearance of double cortin in these cells. This is before treatment, this is after treatment, and I'll jump to the bar which represent the average of the data. We could see first that in 5X FAD mice, there is much less neuron that survive, so much less neuron that express double cortin, and this is this is in 6-month-old mice. After the treatment, there is a significant increase in the number of new neurons that survive. This is in the 5X mice. But we also noticed that in the control mice, there is also an improvement in the number of neurons that survive. So maybe the HBOT treatment can be also used for wild type and also enhance neurogenesis in these mice. So the next thing we wanted to understand is whether there is any effect on the blood flow and blood vessel. And we joined forces with Pavlo Blinder from our institute together with Amos Gdaliau. And we used 2-photon imaging in order to look inside the brain, to look both on the diameter of the vessels and also on the blood flow itself, and also on the plaques that you can see surrounding at least in this case, an artery and a plaque surrounding it. And in the Pavlo Blinder, they developed a method in order to be able to measure the blood flow by measuring the movement of the blood cells in the blood. I will not go into detail, but for that, we designed different experiment. We wanted to be able to come back to the same vessel we imaged before treatment on the same mice and come back to it after the treatment. So we took 6 old mice. We generated the window in the barrel cortex. We did the first 2-photon imaging measuring blood flow and vessel diameters. Then we brought the animal for a 1-month treatment, 1 hour per day, the same thing. And we imaged the same vessels again 1 month later. and tried to look for the same vessel and compared the flow and the diameter. These are preliminary results, but they are quite promising. What we can see in control, so transgenic mice, AD mice, there is some kind of restriction of the vessels. The diameter is going down within a period of 1.5 months. And this reduction in diameter is blocked in the mice that undergo treatment. Similarly, if you look at the flow, there is a reduction in the flow of the blood, the blood flow in the 5X FAD mice. But if we treat them, there is a recovery or actually some kind of an increase. So the effect itself on the blood vessel, we still have to quantify it more vigorously, but it seems that the HBOT prevent the reduction in the blood diameter, in the vessel diameter, and also in the reduction in the blood flow. So if I summarize so far, we can see that HBOT reduces hypoxia in the hippocampus, improves behavior of the 5X FAD mice, reduces amyloid load, and also increases the survival of adult-born neurons. And there is indication that HBOT attenuates the reduction in vessel diameter and in blood flow. At this point, we decided to move to another strain, just to see that it's not an epigenetic of the first one, and we used the triple transgenic mice. The advantage of this mice is it also has a mutation in tau, a phospho-tau mutation. We can also look on this element. Here we took very old mice, so these were 17 old mice. So, and in this case, we decided to treat them only for 2 weeks, including weekend, since some of these mice start to die at this age. So it was quite tough for Ronit, who did the work, and then we did a battery of experiment. And similarly, what we found is that there was a initial stage, there was a hypoxic hippocampus, and following the treatment, the hypoxia is gone. So this is very similar. We did, in this case, different behavioral tests that demand a bit less from the animal. They are quite old, so that either a Y maze or object recognition, and there was an improvement in both behavior after the treatment. We also looked at the phosphorylation of tau, and this is the case before the treatment. So mice without treatment, there is a high load of phospho-tau, and this is reduced significantly after the treatment, just to check that it's not a reduction of tau. So Ronit also did the Western, and she could see that the level of the regular tau is the same, but there is a reduction in the phospho-tau. Similar to what we found with the 5X, and in this case, we look specifically on the subiculum. The plaques in this strain is usually specific to the subiculum, and there was also here a reduction in plaques, both diameter, area, and number of plaques. At this stage, we wanted to go a little bit more detail into the mechanism that might explain it, and we looked at the changes in neuroinflammation and astrogliosis and microgliosis. And the first thing that we looked is the effect of the treatment on astrogliosis, and this is an area of an animal before the treatment or without treatment, and this is the same area or in different mice after treatment. We could see here that in addition to the reduction in plaque, there is also a reduction in astrocyte level, so there is less astrogliosis in this case. We moved to check for microglia, similar to what we heard before, and what we found is that there are less microglia in this area, and the macroglia shape is changed. In order to try to quantify what is the effect on the macroglia shape, and usually, as we heard before, this can recall some changes in the activation state of the macroglia. We use the Weiss neuromat method developed at the Weizmann and quantify different parameters of the macroglia. In addition to a reduction in their number, there was actually an increase in the sprouting and also in the total outgrowth of this microglia. A bit opposite to what we heard before, and we can discuss it later on. So some thoughts that we had. So we saw that HBOT reduces microglia number. We saw that microglia are more sprouted. And this might suggest that HBOT might cause a shift from an M1 microglia, which secrete more cytokine but are less do less phagocytosis, to a M2-like microglia, which secrete less cytokine but are doing more phagocytosis. So in order to do that, we checked for different marker for cytokinesis, for cytokine and for phagocytosis, and we started to look for changes in TNF-alpha, both on the level of the protein and RNA, and there was a reduction in the amount of TNF-alpha in the tissue. Additional cytokine that we checked is IL-1 beta, which was also reduced after the treatment, suggesting that HBOT reduces pro-inflammatory markers. To look on the other side of the equation, we look on the level of anti-inflammatory cytokine like IL-4 and IL-10, and there was a significant increase in their expression, RNA level in the tissue. And lastly, we checked for the effect of scavenger receptors. Scavenger receptors are sitting on microglia and are involved in phagocytosis. And we found that there is an increase both in scavenger receptor A and also with arginase A, both on the RNA level and also on the protein level. So maybe the increase in phagocytotic marker might explain the plaque clearance, while the reduction in microglia and neuroinflammation might be one of the mechanisms that improve cognition deficit in the AD following HBOT. So is there a link between reduction in the number of microglia and reduction in neuroinflammation and improvement in cognition? And a couple of years ago, there were 2 papers from Kim Green labs that show exactly the same. So elimination of microglia improve functional outcome both in regular mice and also in the triple transgenic mouse. So we have at a certain point when you have less microglia which are toxic, if you get rid of them or reduce the number, this can have beneficial effect. And we can discuss it in a minute and relate also to the previous talk. So if I summarize what I said, so HBOT improved cognition disability even in genetically modified mice. It targets several pathways related to AD. It reduces hypoxia, it reduces amyloid load, and it increases neuronal survival. And it also reduces neuroinflammation and might enhance phagocytosis, and might also affect blood vessels. So what we are suggesting that HBOT may present a novel therapeutic intervention of AD. Some of the current work that we are still doing is really to try to characterize more deeply the mechanism. And the first thing that we are now starting is to really examine the effect on synaptic transmission and synaptic plasticity. We want to check whether there is accumulation of oxidative reactive species and to what level, and whether it also enhances the scavenging enzyme. We wanted to understand whether the effect maintained and for how long. In addition, we wanted to check whether we can do something like a dose-dependent study, and maybe in human you can use it, but you don't need 1 hour per day. Maybe you don't need 1 or 2 atmosphere, whether maybe 1.5 atmosphere and 50% oxygen or better, and this might reduce any additional bad effect. And additionally, and relate to the previous talk, maybe we combine the non-invasive treatment of HBOT with gamma wave LED stimulation to modify AD in several directions. So I'd just like to finish by acknowledging a really talented student, Uri Ashery, who did most of the work. He got it for the lab manager who is coordinating everything, and many collaborators that we have during this year. And thank you for your attention.